So I'm going to make a video on the Calvin cycle, or the second part of photosynthesis, and go into a little bit of detail. And just to reference you to some pages, the pages are 395 in your textbook through page 399. All right, oops, 399. And not all of that is carbon cycle, but that is what I need you to look at to supplement your knowledge on photosynthesis. So the Calvin cycle is actually broken down into two parts: carbon fixation and RUBP regeneration. And we're going to talk about what those things are. But if you're curious to know who Calvin is, and you should be, there's a green box on page 397 that you should read called Calvin's Lollipop Apparatus. Read that, see if you can answer the questions in that section, and we can start our conversation in our next class about that experiment. But let's talk a little bit about carbon fixation. So, again, this is in the light independent reaction, which happens. Let me make a new page here. So it happens in the stroma, right? So, of a chloroplast. So I'm not going to redraw that because I think you should already have that in your notes. But the Calvin cycle, or the carbon fixation, is a really important process. This is how we take inorganic carbon, like from carbon dioxide, or excuse me, not even we, right? Plants and algae do this, and some bacteria. Take that inorganic carbon dioxide and bring that into their being, into their structures, and turn that into carbohydrates eventually, right? So that's kind of a really cool process, I think. But this is happening in the stroma of the chloroplast, and in that stroma there has to be this really important enzyme that's called a carboxylase. Carboxylase. And if you remember from class, when we talked about this in the POGL, ASE refers to enzymes, and the first part of this refers to the substrate that enzyme works on, or what its job is, and so it has something to do with carboxyl groups, which I hope you might remember from chemistry. If not, go ahead and Google what a carboxyl group is. And really what's happening here is that there's an enzyme that exists in the stroma in high concentrations. This is a protein that was generated by the chloroplast's own DNA to make this thing, this carboxyl group, this carboxylase. So I'm going to draw a reaction here. It's in your book. I'm going to try to mimic it as much as possible. Over here on the right hand side. So what we have, I'm going to draw, you'll see a 5 carbon. Oh, let me actually do that in a better color. So for biology, what we often do is gray for carbon. Chemists do black, but biologists do gray. I don't know why. So we have a 5 carbon structure here, and we're going to label it ribulose biphosphate. Ribulose biphosphate. Okay, and it's going to become, this is a 5 carbon sugar, it's going to get combined by the CO2 that's being absorbed from the atmosphere. It's got 1 carbon in it, so I guess I should do that in gray. It's a 1 carbon molecule, and those are going to get combined by this carboxylase, and that carboxylase has a pretty cool name. And it goes by ribulose biphosphate carboxylase. So it tells you what it works on and how it's working on that. So ribulose biphosphate carboxylase. Okay, make that arrow a little longer. It also goes by another term, which is short, called Rubisco. Okay, so Rubisco is involved in taking this 5-carbon sugar and combining it with this lone carbon that's being absorbed by carbon dioxide. So now we're going to have a 6-carbon structure. But it's not so easy that it's just going to make C6H12O6 here, because if that was the case and we would have run out of this reactant of ribulose biphosphate, you'd need to replenish that, and that doesn't fit so well with what we would know as a cycle, right? This is a part of the Calvin cycle. So it's not quite so straightforward, and what we end up getting out of this is 2-glycerate 3-phosphates. So the term for this, an abbreviation, which you need to know the name as well as the abbreviation, is G3P. Okay? But we get 2 of them. And each one of those has 3 carbons. So we end up with 2 3-carbon structures. That still accounts for all 3 of our carbons, right? So then there's a series of reductions that happen. Reductions that happen by the NADP and ATP, the NADPH and the ATP from the light-dependent reaction. So this will go down through another set of reactions, and some of the electrons that were in the ATP, 2 ATPs that came out of the light-dependent reaction, are oxidized, so they lose their electrons, and they become 2 adenosine diphosphates plus 2 inorganic phosphates. Phosphate's different than phosphorus, right? Phosphate represents a group centered of atoms around a phosphorus. And also the 2 NADPHs, NADPH, and some protons are oxidized to make 2 NADP pluses. So electrons and hydrogens went into this when we're combined with the G3P. And what comes out of this is a new product called there are 2 of these, 2-triose phosphates. Okay? And they also are 3-carbon molecules. But they have what's been added to them. I'm only talking about carbon, but there are other atoms too, right? So these hydrogens have been added, and some electrons, right? So the number of hydrogens and oxygens, the number of hydrogens is increased, and we've got carbon and oxygen already in the system from carbon dioxide. And in the end, this process is allowing for that all-important ratio that you may remember from 9th grade about carbohydrates, that for the monomers or the building blocks of carbohydrate, for all carbohydrates, there's a 2-to-1 ratio for hydrogens to oxygens. So there's C6H12O6, that's that same relationship. And you see that with every carbohydrate. And that's because for every oxygen that is here, there's hydrogen being added to hit that ratio. But then we need to talk about what happens to these triose phosphates. And that's maybe it's a little more confusing story, but you need to know this set of reactions and the products, and you got rubisco, you've got ribulose biphosphates, you've got G3Ps, and triose phosphates. But the next thing is what's happening to these triose phosphates, because they are going to be used partially to make more ribulose biphosphates, because that's not the end. If these 3 carbons went into C6H12O6, then we would run out of the reactant unless somehow the plant was able to make it. And that's really what we're talking about, is really that these are not all used for making carbohydrates. only a small part of them are, and the rest of it's regenerating into the ribulose biphosphates. So let me open up the next page here. So what we're talking about now is the regeneration of RUBP, right? Ribulose biphosphate. So what's important here is that you understand that, again, things have to cycle through here. So when we have our triose phosphates, if you notice that we have 2 for every carbon dioxide. But we actually need to go through that several times. We need to go through that lots of times, because what happens is that those 3 carbon triose phosphates are going to get converted. So what happens is that we actually need 5 of these for this reaction. So the carbon fixation has to happen several times. We take 5 of them. There's actually a 6th one, right? Because it makes them in pairs. So this is from one CO2, a second CO2, a third CO2. And these 5 are going to get converted. I'll make that arrow a little longer, by taking on some electrons. It's going to become a larger molecule, so it's going to be, the ATP is going to be oxidized into 3 ADPs, whereas these triose phosphates are going to be reduced to make 3 ribulose biphosphates. So these 15 carbon atoms end up making 3 5-carbon RUBPs. But that only leaves these 3 carbons over here to make, to contribute to, I'm going to write this over here, contributes to glucose, to C6H12O6, right? And just to review, this is the result of 1 cycle of the Calvin cycle, this is the result of a 2nd, and this is the result of a 3rd run of the Calvin cycle. But we only still have half, right? This is only 3 carbons, and that's only half of the 6 that we need. So this reaction has to happen 3 more times. So it takes 6 runs of the Calvin cycle to make C6H12O6, or glucose. I hope that makes sense to you. And that's really how RUBP gets regenerated. But what I want you to also look at, I'm not going to go through this because I'm looking at my time. I want you to look through pages 398 and 399, because it's not about the reactions, but really the structure of the chloroplast and how the structure and function are related to ensure that it has a high efficiency of photosynthesis. But also look at the purple box that starts at the bottom of 399 and continues on to 400. And then also look at the questions that come after that and see how well you can answer those questions, so that we can have a rich conversation in class next time.